Back to another breakdown. Here we go again. Yes, sir. Yes, sir. You already know how it goes. We're going to be doing a BB passage breakdown. Okay, this is in the blueprint exam. We'll be doing passage number 1 here. Okay, I'm going to show you guys how to read the passage in a timely fashion. You know, you don't want to read it too slow. You don't want to read it too fast. I'll show you guys how to do that. I'll show you guys how to pick the best answer choice, how to not fall for the AMC tricks. I'm going to show you guys how to do everything there needs to be done to score well on the MCAT. Okay, this is a BB passage, guys. Now, before I break this down, go ahead and read the passage on your own first and answer the questions on your own first, and then hear me break it down. Okay. So I'm going to scroll down here, pause it whenever you need to. Okay, oh, it's kind of a long passage here. All right, so pause it whenever you need to. This is question 1 here. Write down your answer. Question 2, write down your answer. 3, 4, 5. That's it. Okay. So write down your answer and see which ones you got right, which ones you got wrong. Here we go. I'm going to break it down for you guys right now. Cataract generation triggered by lens injury is considered to be driven by the action of the proliferation and epithelial mesenchymal transition of lens epithelial cells as a complication of injury and inflammation. Wow, that's a long sentence. Okay. Not too bad. This is the transition. EMT is the transition. LECs are lens epithelial cells. Okay, when there's injury, the lens epithelial cells go through EMT and proliferation. Cool. The epithelium quickly expands, creating a 100-fold increase in the number of cells. In response to trauma, irritation, or surgery, cytokines and growth factors increase in the aqueous humor and stimulate LECs to proliferate and undergo EMT. Make sense? Okay. This response is significantly diminished in organisms where EMT is inhibited by the inactivation of transforming growth factor TGF-beta, especially TGF-beta 2, the major isoform in the aqueous humor. Okay. We got inhibiting, inactivation, this, okay. So I'm going to highlight EMT is inhibited by the inactivation of transforming growth factor. And we're getting into some details. So I recommend, okay, for you guys to take a quick note. If you need to, do a quick note on the paper or on the horrible marker paper they give you for the MCAT. Okay, make a quick note. So this is what I would make, all right, because I don't want to get lost in the details. So I'm going to go on the whiteboard here. Okay, it's this one. And I'm going to make a quick note. So this would be my paper for the MCAT. My quick note is going to say, EMT inhibited by inactivation of transforming growth factor TGFB. So what I'm going to write is this. When we have a decrease in TGFB, this is equal to a decrease in what? A decrease in LEC undergoing proliferation and LECs undergoing EMT. Okay, so we have a decrease in EMT, which is that epithelial mesenchymal transition. All right. So I just wrote that real quick on my paper. This should take you 2 seconds. Quick, boom, boom. Okay. Let's keep going. During the process of EMT, so epithelial mesenchymal transition, LECs undergo cytoskeletal rearrangement. This is popping out at me, so I'll highlight it. With the addition of a large amount of extracellular matrix proteins such as collagen and fibronectin. Okay. In anterior cataracts, the proliferation and EMT of LECs in situ leads to the formation of opaque plaques just beneath the lens of the anterior capsule. All right. So it's the proliferation and EMT leads to the formation of these plaques. The production of TGF-beta is catalyzed by histone deacetylases. All right. Again, if you guys want to take a quick note here, you could just say increase TGFB by histone deacetylase. All right. Histone deacetylase 1 removes acetyl groups from N-acetyl lysine groups on histone. Once synthesized, TGF-beta is secreted by activated monocytes in the aqueous humor. All right, what's it doing? Who synthesizes it? Activated monocytes. Since monocytes mediate almost 80% of the local TGF-beta, scientists tested the theory that TGF-beta is responsible for the monocytes' impact on the LEC production. TGF-beta responsible, monocytes impact on the LEC production. The number of LECs expressing M1b1, a nuclear protein, was utilized as a measure of LEC proliferation and expressed EMT. Okay, so what's M1b1 mean? M1b1, okay, is a marker to see how many LECs we have. If we have a lot of M1b1, we have a lot of LECs. So if this was my paper, I would say, boom, high M1b1 equals high LECs. Okay. Now to get into the experiment here. Wow, it's a long passage. After simulated injury, monocyte/LEC cultures were treated with an antibody that lyses all but a negligible fraction of available monocytes. A division of these cultures was also treated with a TGFB agonist. A second set of cell cultures treated with antagonists of epithelial cell TGFB receptors TGFBR1 and TGFBR2. So the TGFBR1 and 2 are antagonists. TGFB agonist. We have an agonist and we have an antagonist. What do antagonists do? They go and they inhibit. Okay. Agonists go and they stimulate, they further activate. All right. And here's a table. Again, don't look at the table, don't look at the figures until the question asks for it. You don't want to waste your time going through these, okay? If you want to read, the description here, that's fine. So, results of the study on monocyte lysis, TGF agonist, and TGF receptor antagonist. Okay. Experiment 2. In an effort to determine whether M1b1 is associated with LEC regulation, LEC density was assessed during the course of epithelial growth in wild type and M1b1L cell cultures, null cell cultures. All right, this makes sense here. This makes complete sense why they would do this, okay? They want to see if M1b1 is related to the LEC proliferation, like diminished LEC, whatever, okay? So how do they do this? Well, they say, hey, I'm going to knock out M1b1. I'm going to see what happens to the number of the lens epithelial cells. Okay, I'm going to see now, compare my knockout to my wild type. Okay, if I knock out M1b1, do I have less LECs? If I don't knock out M1b1, do I have more or less LECs? That's what they're trying to figure out, and that's their information here. So, and they have the time here. So we don't look at this only when the question asks for it. If you want, you could read the description here. So growth of LECs after an injury in both wild type and M1b1 null strains. Cool. Long passage, guys. In my opinion, in my honest opinion, this is very, very detailed. So I could see why some people would get lost in the details. All right. That's why for these, it's okay if you guys do a jot real quick. Okay. This jot to be, I wouldn't recommend like going on other people's videos and, jotting down exactly what they jot down. Do it for yourself. Whatever works for you, jot it down. Okay, it has to be quick. I don't want you guys spending a lot of time with this. The point of jotting it down is to for you to go back and look at it and be like, okay, this was this in case the question asks for it. And it helps you to memorize what's going on in the passage. Okay. So do I feel amazing about this passage? I don't feel amazing. I feel good, but I don't feel amazing. And it's okay. So let's keep going. The catalyst for TGF-beta production most likely plays a direct role in what? Okay, the catalyst they told us was the HDAC. All right. I told you guys, if you want, you could write it down. I mean, I remembered it, but if you wanted to write it down real quick, you could have. Okay, so this would be the time where you write down, what the catalyst was, and then you quickly look at it and say, okay, what does it do? So the TGFB is catalyzed by histone deacetylase. All right, this is kind of content review here. Kind of content review, kind of not. All right, you should know what histone acetylation does to chromatin. Okay, you should know that when we acetylate chromatin, specifically at those histone proteins, okay, when we acetylate those, that's going to loosen the chromatin, and that's going to, upregulate gene expression. Okay, it's going to induce gene expression when we acetylate those histone proteins on chromatin. When we deacetylate histone proteins on chromatin, we're going to do the opposite. Instead of activating gene expression, we're going to silence gene expression. Okay, so you should know that from your content review, I would say. And if you didn't know that from your content review, let's say, okay, they tell us that it removes acetyl groups from histone. Yeah, you should know that for your content review. I would say it's a pseudo discrete here, but you could probably get the answer by limiting as well. Okay. Ribosomal inactivation. No, that has nothing to do with this. Transferring acetyl group from acetyl-CoA. This would be histone acetylases do this, not histone deacetylases do this. Okay. Gene activation. Again, this is histone acetylases activate genes. Histone deacetylases silence genes. So the answer is A here. All right. Monocytes, in conjunction with epithelium-derived factors, can act to facilitate which biological process? Okay. You should know what monocytes are from your content review. Monocytes, I would say they're in their immune response, okay, and they can be differentiated into specific macrophages. Okay, that's the role of monocytes. Okay, they phagocytose. All right. Fatty acid oxidation, no, that has nothing to do with monocytes. Okay, fatty acid oxidation has to do with like matrix of mitochondria, those type of cells, breaking down fatty acids. There's nothing to do with monocytes. Transfection, no. That's when you have the vector and you want to put in the DNA in the vector for a specific, DNA library, all that stuff. You can use that. That has nothing to do with monocytes. Okay. Remember, monocytes, they phagocytose. They phagocytose specific bacteria and all that. Lipid synthesis, no. Host immune response, yes. Okay, we want to phagocytose those bacteria, whatever those pathogens are. Okay, maybe display them on an MHC, something like that. Okay. So the answer is D. That's content review. After injury-induced cataract formation has begun, which of the following are least likely to be found in nearby monocytes? Okay. They told us in the passage that after we have injury, okay, we're going to have that proliferation. Okay, and that's going to form the cataract. Now, these monocytes, in order to proliferate and all that stuff, they're going to go ahead and secrete a TGFB. Okay, my bad. So this is what's happening here, okay? And if you forgot, it's okay to quickly go to the passage and read it, okay? Because I forgot a little bit, and that's fine. Okay, so here. When synthesized, TGF-beta is secreted by activated monocytes in the aqueous humor. All right, so I know that TGF-B is secreted by activated monocytes. If something is being secreted by the cell, then I'm going to need a transporter. I'm going to need a transporter to get that protein and transport it out of the cell to be secreted. So I'm going to need TGF-B transporters. The question is asking which one's the least likely. So this is wrong. TGF-B receptors. Okay. Monocytes, okay, they do secrete this. All right. And receptors. What do they need receptors? Okay. This could be it. I don't see why they would need receptors. I mean, if they are proliferating, okay, then it would make sense to have receptors. Maybe that receptor can do amplification and upregulate the proliferation. You know, maybe TGF can bind to a receptor and that will tell the monocyte to go and proliferate further. So that could be it. That could be it. Tight junctions. Monocytes, they would not need tight junctions. Why would they need tight junctions? I don't see why they would need tight junctions. I like the answer C better than B. Why would they need tight junctions? Monocytes, they go and roam in phagocytose, you know? I don't see why they would need tight junctions. So I like this answer better than this one. Cytokines, again, they told us that growth factors and cytokines increase and stimulate, okay? We're going to need those cytokines there. Cytokines help differentiate monocytes too. So we're going to need that. So this is going to be wrong. So I'm left with GGF, beta receptors, and tight junctions. Honestly, I'm going to go with C. C is a better answer choice. Okay. If we need to proliferate, we could possibly use TGF beta receptors. All right. So yeah, I'm going to go with C. Tight junctions. The structure of lysine is shown below. This molecule is best described as what? Easy, guys. You should know this. This is basic polar content review. I shouldn't have to go over this. Given the findings in the passage, which additional result most strongly indicates that the TGF-BR1 and TGF-BR2 receptors regulate TGF-beta dependent epithelial proliferation? Okay, so this we could go through the answers. Treatment with TGF-BR1 receptor agonists resulted in reduced M1B1 detection. All right, let's see here, guys. If we treat with an agonist, that's going to result in increased M1B1 detection. Okay, remember, that's an agonist, not the antagonist. The antagonist is going to reduce M1B1. And this is also shown in the table here. Okay. This is the number of M1B1 positive cells. Okay. When we have no antagonist, we have 42 here. Okay, look at the mouse. 42, injury, no antagonist. When we add an antagonist, let's say we're adding antagonist TGF-BR1, when we add it, we have less cells. We have less M1B1 positive cells. This answer choice is saying that we have reduced M1B1 with an agonist. Okay, with the agonist, let's see what we have. With the agonist, we have increased M1B1 detection. Okay, so this is wrong because it's wrong in the table. Expression of TGF-BR1 and TGF-BR2 was observed to increase during intervals of heightened epithelial cell proliferation. Yeah, that makes sense. Okay. Remember, they want to provide evidence that these receptors regulate TGF-beta dependent epithelial proliferation. If we see both of them increasing during cell proliferation, we can kind of make the assumption that it's the epithelial cell proliferation is due to the TGF-BR1 and TGF-BR2 levels. Okay, so we can kind of make that correlation here, and that correlation would provide evidence. Hey, maybe the epithelial cell proliferation is due to the TGF-BR1 and TGF-BR2. So this is a good answer. C. Genes for several different receptor subtypes were found in untreated epithelium. We care about these receptors. We don't care about all the other different receptor subtypes. Okay, what? That's wrong. Okay, that's nothing to do with this. This is like out of the ballpark here. Okay. Treatment with new TGF-BR3. Okay, I see TGF-BR3. Why do we care about TGF-BR3? We care about TGF-BR1 and TGF-BR2. So I'm going to hit the axe on this and chop it out right away. So I chopped out this one, and this one. And through process of elimination, I'm going to go with B. B makes sense. Okay, it makes sense. So, yeah. Yeah, I feel pretty confident about that. And that's it, guys. So let's go ahead and check if we got everything right. Okay, remember, I'm very confident when I chop out. When you guys chop out answers, be confident. Okay, don't look back at it. So let's see if we got everything right. So let's go back to question 1. Let's see. Oh, this was the first. This is the CP section. This is the cars. So we're going to go to the BB here. Okay. A is gene silencing. We got that correct. This is the explanation. All right. The next one we chose was host immune response. This is correct. We got this correct. This is the explanation if you guys would like. All right. Remember, these monocytes can differentiate into macrophages. All right. Question 3, tight junctions. Here it is. We got it correct again. Confident in my answer choices. That's the explanation if you guys would like. And, oh no, we got another one. Polar and basic. This is pretty much it. I don't need to explain that. And then 5 was B, which is the one we chose. Okay. We got that one correct through process elimination. And this is the explanation if you guys would like. All right. There we go, guys. Make sure you guys, if you guys are interested in working one-to-one with me inside MCAT University, go ahead, go to the comment section of this video, fill out the link to interview. Okay. And if it seems like we're a good fit, then we'll work together and I will make sure, I will hold your hand step by step, make sure you hit that target score no matter what. No matter what, guys. All right. The MCAT is easy. Stop thinking it's hard. Trust me. I know it could be a lot sometimes, but you got this. Okay. Make sure you subscribe and I'll see you guys in the next video.